Hi everyone, my name's Mr. Barlow and welcome to episode 39 of the VCE Biology podcast. This episode covers part of Unit 4, Area of Study 2, and I'll be talking about the way that humans are intervening in evolution. This episode will also include some of the technologies that are used to alter natural evolution and reproduction. So in nature, natural selection occurs to drive evolution. So that is, organisms which have a higher biological fitness value due to their particular characteristics are able to more often successfully reproduce, and therefore they make a greater contribution to the gene pool. So therefore the characteristics that they have are going to be represented in higher numbers in the next generation, and that's basically evolution. But humans can manipulate this natural selection in a whole bunch of different ways. So the first way that humans can alter evolution, if you will, is by doing artificial selection. So pet breeders or farmers perform artificial selection or selective breeding on their livestock. So basically they pick the animals that are going to breed, and they pick them because if they're breeding cows for meat, they pick them because they've got better, nicer tasting meat, for example, and they pick those animals to breed so that their characteristics, their particular characteristics that they're after, are passed on to the next generation. So what they're basically doing is they're manipulating evolution. They're picking the characteristics that they want to see present in the next generation and making sure that only those animals reproduce, and therefore making sure that the species, or the species, part of the species that they control, changes or evolves in a way that they want it to. So when breeders are performing artificial selection on their livestock, they've got a few reproductive technologies that they can choose from. So one of the things that breeders can do to make sure that only the animals that they want to reproduce do reproduce is artificial insemination. So artificial insemination is obviously artificially inseminating a female of the species. So you pick a male of the species which you think has got some really fantastic characteristics that you want to be passed down to the next generation. You then collect some of the sperm from the male of that species and you introduce it into the reproductive tract of the female of that species, and the egg's fertilised by the sperm and reproduction occurs and new organisms are formed with the characteristics that you want. A problem with widespread artificial insemination, if you've got one particular male of the species, a stud cow or stud bull, in fact, is that you can end up basically really decreasing the genetic diversity of a species, and this is obviously not very good. So loss of genetic variation from a species is not a very good thing to happen. But artificial insemination is a way to ensure that if a male characteristic is what you want, you can artificially inseminate lots of females. Another technology is multiple ovulation embryo transfer, or MOIT. So in this case, you can basically increase the reproductive potential of a female. So artificial insemination increases the reproductive potential of a male, and MOIT increases the reproductive potential of a female. What happens is a female with the particular characteristics that the breeder is keen on reproducing is given a hormone, follicle stimulating hormone, so that she ovulates heaps, so produces heaps and heaps of eggs. Her eggs are then fertilised, and they can be fertilised by a male of the species which has particular characteristics, and then the newly formed embryo of the egg that is desirable with the sperm that has the genetic material which is desirable is removed from the lab and put into the reproductive tract of a different female, so that lots of different females give birth to new organisms, but all those organisms have been fertilised by the same male and the same female which have particular characteristics that are desirable. Another technology available to breeders these days is basically sex selection through sperm sorting. So if you collect the semen from a stud bull, for example, you can then treat all of the semen with a fluorescent dye and then separate which sperm have got an X chromosome in them and which sperm have got a Y chromosome in them. So the sperm which have an X chromosome, obviously if they fertilise an egg, it'll result in an XX organism, or a female. And the sperm with a Y chromosome in them, when they fertilise an egg, it'll result in an XY organism or a male. So breeders can basically pick which sex of the species they want to occur when reproduction happens. So if you want a whole bunch of females, you pick all the sperm which have an X chromosome. If you want a whole bunch of males to be born, you pick all the sperm which have Y chromosomes in them. So that's the ability to sex select by looking at the sperm. You can also do artificial selection in plants, and so plant sexual reproduction involves pollination. So artificial pollination is basically the plant version of artificial insemination. If you've got a plant which has particular characteristics you're after, you get its pollen and you fertilise another plant which has the same characteristics you're after, and then you can control the evolution of the plant in a very similar way. So that's artificial selection, and a whole bunch of ways you can do it. Another amazing way that humans are currently intervening in evolution is cloning. So normally in sexual reproduction, two different individuals, two parents get together, and their genetic material contributes equally to a new offspring. And it's a mixture of those two parents. But in cloning, what happens is one organism is cloned. It's replicated precisely to form another organism, which is a clone, which is an exact copy of it. So there's a couple of different ways that cloning occurs. The first type of cloning I'll talk about is embryo splitting. So what happens here is that when an egg is fertilised by a sperm, a zygote is formed, and then that cell replicates and replicates and replicates a few times to form a blastocyst, which is basically a small collection of cells, but they're all stem cells, so they can all form different parts of the organism. But in embryo splitting, what you do is you take the blastocyst at maybe the 8-cell stage and split or separate all 8 of those cells into, apart from each other. So then you've got 8 different embryos, and each embryo can form its own new organism. So you get those 8 identical embryo cells and put the 8 cells into maybe 8 different surrogate mothers. But then all of those surrogate mothers will give birth to a new organism, which is a precise clone of all the others. So embryo splitting is a great way to basically increase the reproductive potential again of a particular male and female combination. So you can get a whole bunch of different surrogate mothers to carry offspring of a particular organism with a particular genetic makeup that you're after. And again, you're altering the evolution of the species. So another way to clone an organism is by nuclear transfer. And this is how Dolly, so there was a sheep called Dolly, which was the first mammal which was ever successfully cloned, and it was quite a breakthrough at the time. So basically what you do in nuclear transfer is you take a cell from the organism you want to clone, basically any cell, and then you just take the nucleus, which has the DNA or the genetic material, out of that cell. So you get the genetic material out of a cell of an organism that you want to clone. Then you get an egg cell from, basically any other organism of the same species, and remove the nucleus from that egg cell, and that's called enucleation. So you get the donor genetic material from an organism you want to clone, and then you get basically a blank egg cell, so an enucleated egg cell. Then you transfer the donor nucleus, the donor genetic material, into the enucleated egg cell. And then, so you've got a cell again, and you've got a cell again with genetic material in it, but it's different genetic material. Then you transfer that egg, the developing embryo, into the uterus of a surrogate animal, and then in an ideal world, that genetically modified embryo develops in the organism and is born, and it's a carbon copy, a clone of the original donor animal where the donor nucleus or donor genetic material came from. So that's cloning by nuclear transfer. The success rate is actually very, very low at this stage, but it's really quite amazing that we can do it. So another way to interfere in evolution is we've now got the ability to transfer genes, transfer stretches of DNA between species. So in nature, cats and dogs can't reproduce. If you've got a cat which has got some really fantastic characteristic that you'd love the dog to have, they can't reproduce. So they can't reproduce to form a viable offspring. And that's because they're different species, and basically genes can only be transferred from parents to offspring within the same species. They can't cross the species barrier. But genetic engineering actually evades this natural limitation. So geneticists now have the ability to transfer genes between species. And organisms that have had any foreign DNA inserted into their genome are called transgenic organisms. And so, this is really useful. A good example is that we've created transgenic cows. So cows which have got genes from different organisms in their genome, so in their DNA. For example, some cows have been genetically engineered to produce human proteins in their milk. So those human proteins can then be isolated from the milk and used in the medical fields. It's quite amazing. The way that genes are transferred from one species to the next is basically you isolate the gene. There's a couple of different ways, but one way is you can transfer the gene using a retrovirus. So you can put the gene of interest, insert that into the genome of a virus, infect the organism that you want to become transgenic with that virus. That virus will then become part of that organism's DNA. And there you go, you've got a transgenic animal. It's just amazing. So the fact that we have the technology to alter the genetic material, alter the genome of an organism, brings up the possibilities of gene therapy. So gene therapy is basically the insertion or alteration or removal of genes within an individual's cells to treat a disease. So if someone's got a genetic disease, now we're looking at ways that we can treat that genetic disease by, removing the gene or replacing that gene with a fully functional version of the gene. For example, the disease cystic fibrosis is caused by a mutation in the gene for the protein cystic fibrosis transmembrane conductance regulator, or CFTR, but it's just one gene. So if we could somehow figure out a way to, we've made some progress, but if we could figure out a really reliable, foolproof way of fixing that mutation or replacing that mutated gene with a fully functional version of the gene, we could cure cystic fibrosis, and we do it by gene therapy. So in discussing the modification of an organism's genes or genomes, the topic of stem cells should come up. So stem cells are these really special cells. They're cells that can reproduce via mitosis and produce more stem cells, but any one stem cell can in fact differentiate to become a diverse range of any specialized cell types. So one stem cell can differentiate to become a nerve cell, or it can differentiate to become a muscle cell or a skin cell. So one stem cell, it's like a blank canvas, and it can differentiate to become any other type of cell. Importantly, stem cell therapy has the potential to basically dramatically change the treatment of human diseases, because if you've got disease and you've got some faulty or some dead cells, you can replace those dead cells with stem cells. So, for example, what they're trying to do at the moment with stem cells is hopefully one day they'll be able to replace nerve cells in people's spinal cords who've got paraplegia or quadriplegia, so that they'll be able to walk again. So it's just the potential for stem cell therapy is amazing. Think of any cell that's not working quite right in you, just get one of your own stem cells and bingo, make it work again. So in talking about stem cells, there's actually a few different types of stem cells. There are totipotent, pluripotent and multipotent stem cells, and that basically refers to their power or their potency to produce a range of different cell types. So totipotent stem cells can give rise to all cell types of an organism. Pluripotent stem cells can give rise to most cell types of an organism. And multipotent stem cells can only give rise to certain cell types of an organism. So multipotent stem cells, they're not really as powerful to produce heaps of different cell types, whereas totipotent stem cells, they can produce any cell of the organism. Think of any specific differentiated cell that you can, and a totipotent stem cell can end up differentiating to form that type of cell. So if a scientist wanted to isolate some stem cells, they could isolate some pluripotent stem cells by basically getting a blastocyst, and they'd be able to get embryonic stem cells. So embryonic stem cells are pluripotent. If you wanted to get some multipotent stem cells, these are adult stem cells or somatic stem cells, and these can be obtained from various places within the body. So from, example, within bone marrow. So, scientists are able to isolate stem cells, and they can get ones with different levels of potency. So we can actually isolate pluripotent embryonic stem cells, but you need to get an embryo to isolate them from. So if a person has a disease or a disorder and they'd like to cure it by using stem cell technology, then therapeutic cloning needs to take place first to get some stem cells. So therapeutic cloning is basically the creation of an embryo that is genetically identical to the patient, the person who's sick. So one of the patient's cells is fused with an enucleated egg cell, so an egg cell which has had the nucleus removed, and their genetic material is put into that egg cell. That egg cell will then develop into an early embryo. So that early stage embryo, or it's actually called a blastocyst, can be a source of pluripotent stem cells. But you can see actually what you've done there by getting an egg and putting some DNA into it and then starting the development of a human, so getting an embryo and forming a blastocyst, you've created a person, you've created a human being. So the creation of artificial embryos for therapeutic reasons creates heaps of ethical issues. If someone's sick, is it okay for them or their doctors or geneticists, is it okay for them to create a whole new person only to kill that person, to use their genetic material or their cells to fix the original person? Is that okay? I don't know. So these are some of the ethical issues that come up with stem cell research. Now, as I mentioned earlier, humans have and still do alter the evolution of heaps of organisms, so particularly livestock and crops. We're basically trying to direct their evolution so that they can be more useful for us. But we've also got technology which alters human reproduction and therefore human evolution. So there's a couple of different things we can do. For example, we've got the technology to stop conception. So, when two humans reproduce and they conceive a new organism, that's basically when sperm meets the egg. It's the production of a zygote. That's conception, but anything that you do to stop conception is called contraception. And, there's heaps of different methods of contraception. There's condoms, there's the pill, there's IUDs. So that's one form of human reproduction. So it's stopping humans from giving birth and therefore changing their biological fitness. We've also got the ability for couples to overcome infertility. So we can intentionally make couples less fertile by contraception, and we can intentionally make couples more fertile by using various technologies too. So a couple of the ways that we can make two humans, male and female humans, more fertile, so increase their biological fitness, is that we can increase a woman's fertilisation by using donor insemination to increase the chances of a woman reproducing. We can do IVF, or in vitro fertilisation, so that's we can collect the eggs from the ovary of a female, and we can collect the sperm from the testes of a male and fertilise the egg with the sperm in vitro or in glass, and incubate the eggs for a bit, and then later transfer that developing blastocyst into the uterus of a female, and then that female can give birth. So maybe she was infertile before, but IVF can basically make an infertile woman, give her the possibility to reproduce. If a woman is unable to carry a child to term, then a surrogate can do that job. So you can get a egg from a female and then fertilise it in vitro, but then transfer that fertilised egg into a surrogate female. So then another woman would give birth to a child with the genetic makeup of the original infertile female. Another way we can overcome infertility is something called gamete intrafallopian transfer, or GIFT. And this is basically where you get the gametes, so you get the sperm and the egg cells, and then inject both of them into the fallopian tubes of the female, and then hopefully they'll meet each other, and the egg will be fertilised, and then reproduction will take place. Another type of technology used to aid human reproduction is intracytoplasmic sperm injection. So this is basically, you get an egg cell, and then you get a sperm cell, and you inject the sperm into the cytoplasm of the egg cell, so that you basically artificially fertilise the egg cell. And then this fertilised egg cell goes through IVF, so it's incubated for a little bit in the lab, and then it's transferred to the uterus of the female for regular development. So, we're also right now, and this is also arguably an ethical thing, we're intervening in human reproduction, where some people are stopping themselves reproducing, so decreasing their biological fitness. Other people who normally would not, or naturally would not be able to reproduce, are reproducing and increasing their biological fitness. So we are certainly interfering with evolution. And that brings episode 39 of the VCE Biology podcast to a close. I'm Mr. Barlow, and once again, thanks for listening.